NAPA is a powerful protein microarray platform that can be used for the study of protein activity and function in an unbiased, high-throughput manner. The proteins displayed on NAPA are produced in a human-based expression system using human-derived ribosomes and chaperones in order to improve the likelihood of natural folding and activity.
The use of NAPA arrays for the screening of kinase inhibitors provide a new platform for the test of new drugs and clinically relevant kinase mutations.
Protein microarrays generated by NAPA methodology can be used for many distinct applications, including biomarker discovery, protein-protein interactions, substrate identification, and drug screening.
Perform quality control steps along the way. Test the quality of your DNA preparation, the DNA microarray, and the protein microarray. If in any step the quality is not good, just start over.
Demonstrating the procedure will be Lisa Miller, a technician in my laboratory.
To prepare bacterial growth for in-house high-throughput mini-prep, first, inoculate the bacteria in the agar plate in a 96-well format and let it grow for 16 hours. The next day, fill each well of a deep well plate with 1.5 milliliters of terrific broth medium supplemented with antibody ampicillin. The antibody depends on the selection marker on the plasmid. Then, sterilize a 96-pin device with 80% ethanol and flame. Use the sterile 96-pin device to pick colonies from the overnight incubated agar plate and submerge into the wells of the deep well plate to inoculate the culture. Cover the block with a gas-permeable seal and incubate on a shaker for 22 to 24 hours at 37 degrees Celsius and 300 to 800 RPM. After that, pellet cultures and resuspend the cultures according to the manuscript.